But I guess maybe it's a good idea to get back to the basic problem. What do we actually need to do in order to understand whole brain emulation? Because the problem is stating that we want to copy a human mind from a brain, that leaves out a lot of details. If you just state it like that, we can't make any kind of predictions whether it can ever be done or not. So we need to consider actually what kind of technology to do. So we organized a workshop in Oxford. So at this workshop, we banned all philosophical discussions about would a copy of my brain actually be me? Would it really be conscious and things like that? We only wanted to get to the technical details. So we had computer scientists, neuroscientists, microscopy people talking and explaining what they knew. So basically, the problem consists of three parts. You need to scan the brain somehow. You need to interpret that scan in some way, and you need to run it on a computer in a simulation. So you can imagine that the scanning method could be non-destructive. I have a reason to believe that's not going to work, because when you think about the resolution you need to scan the brain, it's most likely going to be done on the cellular level, for reasons I'm going to get into later. That means that you're going to need to scan something on the scale of less than 0.1 micron over a distance of several centimeters if you want something on the outside of the head. The problem is that just the blood flow in the brain makes everything wobble several hundreds of micron every beat of the heart. And not to mention, of course, that things are moving around in a slippery way, etc., etc. Terribly annoying on its own. So I think there are pretty good reasons to believe we can't do that kind of scan. You can imagine something like a magnetic resonance camera. And if you have a really, really strong field, you get better resolution. So that might sound like a solution, except that they are based on the notion, well, water is diffusing about 0.5 micron over a normal scan. So if you actually try to do that, you can't get a better resolution. So there are kind of technical reasons why we can't do that, even with infinitely strong magnetic fields. There might be other ways, but it seems very, very hard to do that. So we concentrated on, well, let's see if we can scan the brain in a destructive manner instead. And suddenly things look much better. So basically we need to figure out what resolution we need to scan the brain on. And we have a kind of list of several levels. At the top level, it's essentially that in order to make a copy of a brain, we need to understand the functions done by different brain areas and their connectivity. So that would be almost like a traditional artificial intelligence model. Then you can imagine something like cortical mini-columns. They're the actual computational units. So we only need to know their properties and how they're connected. Then you can imagine that you would need individual cells, but you don't really care about the details. They're just simple like an artificial neural network. Then you can imagine a level where you need to have electrophysiology like we're running in current computational neuroscience. You have a compartment model where we model the electric currents going through the membrane, some of the chemistry. And then you can imagine even more elaborate ones. You can have models where you need to take detailed biochemistry into account. We might want to have models where we keep the kinetics of different enzymes into account, all the way down to models where we model each individual atom and how they're vibrating and moving. And then you have the ultimate level where you do a quantum mechanical simulation of the whole wave function of the thing. That level is going to require quite a lot of computing power. Essentially, it's entirely infeasible to actually do, I think. Even though doing a classical mechanical simulation of molecules is going to need a lot of computing power. But the cool thing is we actually understand the laws of how atoms are moving very well. We don't need to know anything at all. If I just had all the data and a sufficiently big computer, I could write the program relatively easily. On higher levels, though, the ones where they require more understanding of what's going on in a neuron, for example. What chemicals are there? How do they interact with each other? So I need more knowledge, but I also need less computing power. So the really abstract model, I'm not going to need much computing power, but I'm going to need to understand the brain quite a lot. So our idea was that we don't know which level we need to run this on, but it's pretty likely that our understanding is slowly going to be creeping up. We're going to be able to model molecules much earlier than we're going to be able to model a complete biological neuron. But it's going to take a while before we get enough computing power. So computing power is kind of going downwards. We're getting the ability to simulate more and more complex systems, and understanding is creeping upwards. We're actually understanding more and more complex systems. Eventually we're going to meet. And if that level is high enough, we're actually going to be able to run something. So we had a kind of show of hands among the delegates. what level they believe that we would need to work on. And most people believe the electrophysiology, a kind of compartment model, would be about right. We don't know that. This is actually an interesting research problem we need to solve. We're thinking about ways of setting up experiments to test that. And one of the best ways of testing it is, of course, to run small animal emulations and see whether that is enough detail to get the hand to actually run and so on. So in order to get electrophysiology, what kind of resolution would we actually need? And it turns out to be about, we guess that 5 times 5 times 50 nanometer resolution. The reason for that is that's going to be able to detect the finest axons and dendrites. And we're going to, with a pretty good margin, actually. So assuming you can get all relevant data out of those pictures, that should be it. The problem is we might need to do a lot of chemical data, which we have a less good idea about. And that turns out to be tricky. But at this point, the microscopic people were giving us some pretty good news. Getting that kind of resolution, you can do that rather easily in an electron microscope. You can even do it in 3 dimensions with various setups where you slice off the upper layer, do a new scan. And you can do this using both various things where you have a little microtome, cutting with a diamond edge, or you can use various lasers or particle beams, and there are other tricky solutions like Kenneth Hayward's an uploom, which is a wonderful construction that kind of uses a lathe to lay out a long strip of tissue on a metal tape. So that way it becomes easy to manage, and essentially you can scroll that tape back and forth under a microscope. So he's envisioning taking a small piece of tissue and turning it into a library, essentially a disk, like a typical silicon die you use when you make microchips. So it can be managed by the same kind of robot we use for microchips, and the same kind of microscope we use for microchips, so you can buy that off the shelf from Intel. So that way you could keep the prices down quite a lot and have this library where the tissue itself is actually a documentation of what's going on in there. The interesting problem is, though, can we figure out, for example, whether a synapse is excitatory or inhibitory from an electron microscope pictures? Well, we don't know that yet. This remains a very deep question. There are some reasons to believe we might be able to do it, but there might also be reasons we can't do it. Fortunately, at Stanford, we developed something called an array tomography microscopy. So that means we get a lot of little tissue slices. I think it's about the same thing as the Athlon. And then you use an immunostain to color the tissue where certain interesting chemicals are. Now, you can image that only using a normal optical microscope, which doesn't have enough resolution. It won't tell you exactly where the synapses are, but you can at least see roughly a synapse. You can make that one out, and you can see what chemicals are in it. And once you've taken a picture of that resolution, then you remove that stain and add a new one for other chemicals. And then you continue this. You can apparently do about 20 chemicals. And then finally you scan with an electron microscope. So now you've got a rough map of the chemicals where they are, and a fine map of exactly where the cell walls are. So that seems to suggest that if electrophysiology and some of the chemistry is the resolution we need, we're likely to be able to do it. We haven't really got the tools to do the really complex molecular structures, the protein complexes are needed. But at least down to that level, we seem to have a technology that would work, except that it needs to be scaled up to an enormous industrial scale compared to the present. So that brings us over to the really tricky part, the part where we know we don't know how to do it yet. And that is interpreting the scans and turning them into a simulation.